I would like to introduce Dr. Diane Simeone, who is a professor here in the departments of surgery and pathology. She's also the Associate Director of Translational Research at Perlmutter Cancer Center and the Director of the Pancreatic Cancer Center. So let's welcome Dr. Simeone.
Very excited that you're all here tonight. I think we're going to have a fantastic session. We're all going to learn a little bit more about pancreatic cancer. I just want to start off that this session was going to be co-run by myself and Victoria Mannix, who's actually the Chief Medical Officer at PanCan, but unfortunately she's ill. So I'll be covering the material of this session, which is going to be on precision promise. Just as a brief outline for the evening, we'll have the topics kind of broken up to three areas. The first will be precision promise, which is a new paradigm in doing clinical trials for pancreatic cancer. The second session will be focusing on new ways to think about using immuno-oncology in pancreatic cancer, and then the third session will be on genetics and elevated risk. So I'm going to show you here is data we all know. Pancreatic cancer has been a very tough one to deal with. Still a single-digit survival rate, and despite a lot of work in the area, I still think we don't have the tool set we need to combat it. There's been data published that shows it's projected to become the second leading cause of cancer death in the United States by 2020, and the incidence continues to increase and rise over time. Now, there are lots of people that are doing important work in pancreatic cancer, but if you view it from the perspective of a pancreatic cancer patient, the pace and the scale at which things are happening aren't happening fast enough. And I would put out there that our vision should actually be to transform the prevention, early detection, and treatment of pancreatic cancer with the aim of increasing the 5-year survival rate to 50% within the next 10 years. Now, I've been told that's too ambitious a goal, but I'll beg to differ. I think that we have to aim high if we're going to get where we need to be. So why haven't we hit that goal yet? What are the challenges we face? As a clinician scientist, I work in the lab and in the clinic, and I can tell you there are things that we need to address to really move forward. First is compelling research doesn't always move to clinical trial, and there can be a disconnect sometimes between scientists and clinicians. The timeline to get from an exciting scientific discovery to a treatment for a patient is too long. Our system is not integrated sufficiently to execute that. Access to new therapies for pancreatic cancer has actually been pretty challenging. It's been a disease that's really been in the shadows for patients, and it hasn't really garnered the interest of the pharmaceutical industry that it should have. And unfortunately, inherent in academia are barriers, institutional barriers, individual barriers to sharing data. The incentive system rewards individual achievement instead of collective group activity. And what I would propose is that incentive system needs to change. Obviously, a key important point is we don't have an early detection test, and I think one of the reasons is we haven't adequately brought all the scientific power that we can to the table in the space of emerging technologies. So if you look at all these issues, what I'm really going to focus on in the next few minutes is tackling these right here and talking about how we can think about going forward in a different way. So I'm always a very optimistic person, and when you look at our challenges, what I would say we're actually in a very favorable position currently to make an impact in pancreatic cancer in a way that we have not been able to do so previously. Why is that? Well, first of all, the science and the scientific community have grown substantially over the years, and we have a much larger body of researchers and a much more complex understanding of the biology of pancreatic cancer. We also now know that not all pancreatic cancers are the same. They're not all in the same bucket, but actually tumors can be quite distinct. and that specific treatments may be most appropriate for different molecular immune signatures or may depend on the host response of the patient, and we need to think about that much more deeply. And then thirdly, we have a much larger arsenal of therapeutic agents at our disposal, and we certainly are likely going to have unique combinations of drugs to really go after pancreatic cancer because it's been much more resilient to treatment than other cancers thus far. So there are lots of therapies out there, lots of approaches. Targeted molecular therapy, immunotherapy, ways to disrupt the scar tissue or the stroma in the tumor, and then other approaches. And so how do we go about figuring out how to go after pancreatic cancer from a therapeutic standpoint in an intelligent way? Now, if we look at where clinical trials are at for pancreatic cancer, I would say they're not working as effectively as they could be to change the course of this disease. The most compelling science isn't moving forward to clinical trials. We talked about access being limited, and the NIH budget for clinical trials has been cut. The average timeline from the presentation of a concept for a clinical trial to actually starting it is an average of 2 years, which is too long. We need to work on sharing data. We don't have good predictive biomarkers, and only about 4% of patients actually go on a clinical trial. So if the current strategy is not working, then let's change our strategy. So how do we do that? I'm going to propose that we should develop a new ecosystem for doing clinical trials in a more impactful way. So we've had a lot of discussion about this across the clinical and research community, and we, in fact, presented a concept to the Pancreatic Cancer Action Network to try to revamp how we do clinical trials. If we could wipe the slate clean and say, how should clinical trials be done like we want them to be done, what does that look like? And from those discussions is born out this new clinical trial paradigm called Precision Promise. And to the Pancreatic Cancer Action Network's credit, they've made a commitment to support this initiative for $35 million for the first 4 years. So if that slate is wiped clean, what should be the basic tenets of the clinical trial design? This is what we would put forth. We should have a novel model of cooperation. Trials should be rigorously vetted by scientific teams. I'm going to talk to you a little bit about adaptive clinical trial design and make the case that we should do clinical trials in a different way. They should be modeled differently. One thing we have really lacked is deeply studying every patient who is on a trial. We have not routinely characterized patients' tumors. We haven't done biopsies during treatment to really understand why things work or why they don't work and learn. We have to have a learning algorithm that's now in place. To be part of Precision Promise, you have to agree to openly share your data. So with all this, the Pancreatic Cancer Action Network had an independent process of soliciting invitations from centers and chose 12 centers from around the country to start off to do this consortium, although over 45 applied, and the goal will be to increase and expand this effort as additional funds come on board. We've used a private funding model to do Precision Promise because we thought that the pace of which we needed at which we needed to accomplish this was going to be best done with private funding, but we're certainly open to working with the NIH as we move forward. I'm going to show you a little bit about the design. If you look in total, that we have 12 new clinical trial innovations with Precision Promise, and we're working with the FDA as quickly as possible. This is scheduled to be open in April of 2018. Now, we talked about limitations of drugs. With this consortium, we've actually been very successful in building out a partnership with the pharmaceutical industry in a way that we've not been able to before. And to feed into all of this, we need to have a matching translational grants program, and the Pancreatic Cancer Action Network has that in place. And of course, I want to also acknowledge the Lustgarten Foundation, who's here today, that also do a terrific job funding basic and translational work along with clinical trials for pancreatic cancer. The most important part of this is we should put the focus of the patient at the center of every single decision we make. So this was the mission statement we came up with: to transform outcomes for all pancreatic cancer patients through a research and clinical trials platform that creates a culture of collaboration and learning among clinicians, researchers, and diagnostic and drug developers, always putting the patient at the center of every decision. We are going to establish new goals and guidelines for clinical trials. All patients should get pre- and post-biopsies, again, to learn why things work or why they don't. Scientific correlative studies will be deeply embedded into every single clinical trial, including comprehensive genomics, immune profiling, tumor biomarkers, and liquid biopsies. During the entire process, we need to have extensive crosstalk between researchers, clinical trialists, and the pharmaceutical industry. And I would tell you, we've actually learned a lot as we designed this from our pharmaceutical partners. And with the novelty of what we've developed here, we can continue to think very innovatively about different ways we can move forward. But our goal is to have patients have multiple clinical trial options or multiple shots on goal. Now, when we started off this process, we looked across the scientific landscape and decided to focus on 3 specific therapeutic areas: stromal disruption, DNA damage, and immunotherapy. Although I would say that as we're moving forward, we're very receptive to all clinical trial concepts. We are establishing a master protocol. I'll talk to you a little bit more about that. An important part of all of this is we actually have deep supportive care research embedded in the clinical trial design. How do we best treat pain? How do we best give nutrition while patients are being treated? How can we treat cachexia more effectively? These are all things that we're going to be studying as well. So here's the slide just showing the 12 sites around the country that were chosen. Here in the New York area, it's NYU and Sloan Kettering, but you can see there's representation across the country. And it'll be launching, as I mentioned, 2018. This has been, from my experience, an unprecedented effort nationally. We have had hundreds of individuals around the country across most of the major institutions involved, well beyond the names listed on this slide. We have a national steering committee, which I have the privilege to chair, and members across institutions around the United States. We have Vince Bacozzi, who heads the Clinical Trial Consortium. We have a Clinical Trial Design Committee, a Biomarker Committee, a Supportive Care Committee, an arm selection committee that vets all the different clinical trial concepts and picks the concepts that have the most promise, and an operations committee. We had very extensive discussions about what the design should look like, getting a lot of diverse inputs, and decided on an adaptive multi-arm platform trial. So what the heck is that? So a platform trial is a trial in which you can study multiple therapies in the context of a single disease in a perpetual manner, with therapies allowed to enter or leave the platform on the basis of a decision algorithm. This kind of approach for clinical trials is pretty new. It hasn't been routinely used in cancer or, in fact, in many diseases. There are just a few isolated examples of this kind of design. But what I can tell you is the leadership of the FDA is very much on board with moving forward in this direction, as shown here on this slide. For those of you who want to learn a little bit more about this kind of clinical trial design, I encourage you to read this article that was published in New England Journal this summer by Janet Woodcock at the FDA, highlighting how we can get so much faster information and move further along using a master protocol to study multiple therapies at the same time. So what are the benefits of going about it this way? You can evaluate multiple therapies simultaneously. We have a common control arm, and we can have 5 or 6 or even more experimental arms all running at the same time. So far fewer patients are in the control arm, which is standard of care. We share that control across all the arms. The trials actually need to be are much smaller than traditionally designed. And the way the platform is run, it's statistically modeled such that if a therapy is working, more patients will be enrolled in that arm, and then if an arm is not working very well, fewer patients will go into that arm. So poorer performing arms are dropped faster. We also currently in our field have no effective predictive biomarkers for therapeutics, and we have to start somewhere. This is going to be a way for us to rigorously develop and validate biomarkers. And it's going to be a much more efficient use of resources. The typical phase 3 clinical trial costs about $40 to $50 million to go through Precision Promise as an arm, to go from a beginning of phase 2 to FDA approval is going to be in the ballpark of about $5 or $6 million. You can see we're going to be able to do much more work for less amount of money. The basic overall design is a primary endpoint of overall survival. We're going to do an adaptive randomized design. I'm going to just show you an example in a slide or two what that looks like. Many experimental arms will be screened, and in the phase 2 part of it, the maximum number of patients per arm will be 100 patients with a minimum number of 20. So if something really doesn't work, only 20 patients will receive that treatment, and then we'll move on with something else. If we have something that works, then it'll just flow seamlessly into a phase 3 clinical trial with a maximum number of 75 patients. Right now, if you have something that works in phase 2, you have to pause, the drug company has to raise a lot of money, and the phase 3 clinical trial is done, which is very laborious. So again, fewer patients have ever been required to get to FDA approval to get treatments that can be available for every single patient. Now, while we're initially going to start out with overall survival as the endpoint, we are going to model out having surrogates for survival, be it tumor markers like CA19-9, tumor imaging, or other blood or tissue-based biomarkers. And very importantly, we're going to have a wealth of data, well-characterized patients with clinical, scientific correlative data and outcomes tracked longitudinally that basically has never existed before. So the platform is as follows. Clinical trial concepts are vetted by the Arms Selection Committee, and through a vigorous process, then arms are chosen. There's the phase 2-3 clinical design, deep study of every patient, and if something works, there'll be an accelerated path to FDA approval or fast failure of non-effective therapies. The underlay of this is supportive care research. We actually are innovating in the financial modeling of how to do this kind of platform. We're going to deploy patient-reported outcomes as part of the readout of the clinical trials. Large-scale genomic analyses will be performed along with comprehensive biomarker discovery. And this is just an example of the process that the Arms Selection Committee goes through to figure out which arm should go in. Concepts are presented along with the agents that can be utilized to target these different areas. The committee does an extensive literature search and has working group meetings. Just in the initial process, we evaluated 74 potential arms. Then we have discussions with industry and really dive deep into the data. And ultimately, through this initial process, we have chosen 3 experimental arms that are going to row through Precision Promise. Although I will tell you there are multiple other arms that are currently in the discussion phase of, with our pharma partners. We can't reveal these arms yet because they're in the midst of contracts with the pharmaceutical companies, but as soon as those contracts are finished, we'll be able to release that information. This has been a pretty eye-opening part of the process for all of us, is by building out this kind of consortium and platform, we have leveraged ourselves in a way we never have been able to before to interact with the pharmaceutical industry. Here's an example of a number of companies that have financially contributed to the platform and are offering to pay for the clinical trial either drugs or arms that go in. In fact, several large companies are providing access to their entire portfolio of drugs, which is quite remarkable. We're also in discussion with at least 8 other additional pharma partners about formalizing a partnership. So let me just take you through this slide for a minute because it gives you a sense of how this kind of platform trial can run. Patients will come into the system, and we expect that thousands of patients will flow through Precision Promise. There's continuous screening, comprehensive tumor analysis, again, deep genomic analysis, immune profiling, along with different biomarkers, adaptive randomization. There will be standard therapy arm or arms, and then a number of different experimental arms. If, for example, you look at experimental arm 2, patients will be entered in this arm, and if it stops for futility, patients who are in this arm will have the opportunity to be re-randomized to another arm, again, giving them multiple options throughout their journey. If you look, for example, here at experimental arm 3, which had a positive phase 2 readout, it can seamlessly flow into a small, focused phase 3 clinical trial with no more than 75 patients to get to FDA approval. And what you can see here is this is something that a kind of platform that just goes on in time, and as new experimental arms come on, they can be added into the system. Now, one thing we also realized is some of the really exciting scientific concepts out there weren't quite ready for the phase 2 3 platform, and so how do we get them from the lab into this space? And so what we want to do is really build out the preclinical evaluation phase, and we are going to build a phase 1B subnetwork so that we can really push arms that we think are exciting into the platform. In addition, while the platform is going to start with patients who either have naive or previously treated metastatic disease, we are working now to extend to locally advanced pancreatic cancer. So in conclusion, what I've shown you is really a new national strategy, which we've put in place to make significant advances in clinical trial options for pancreatic cancer patients. And this new cooperative model aligns key stakeholders, and there are many in the process: patients, families, researchers, clinicians, foundations, pharma, FDA, and even payers. Aligns us all in a unique way to increase the pace and scale in which we can impact change. And importantly, we really want to emphasize having cross-mingling of expert scientific teams, which will allow unique combinatorial approaches to be best tested in a rapid and efficient manner. So overall, this will result in smarter clinical trials embedded in strong preclinical data in our future. Now, and all that being said, what are the real opportunities we have here? You know, I presented this at the beginning. I think that this kind of effort can actually be transformational for our patients and can get us to a place where we could have a 50% 5-year survival. The pharma consortium that we have developed not only provides agents for clinical, but also importantly preclinical testing. A lot of times researchers can't actually access these drugs in the lab to test them. You could see how a platform like this could be expanded to also do clinical trials to test biomarkers for early detection or even prevention in high-risk individuals in the future. We expect the network will expand to 30 to 40 sites over the next 3 to 4 years because the goal is always to be inclusive, and we will be considering innovative funding models to do that going forward. And then lastly, this large-scale effort, we'd love to link it to other efforts by pancreatic groups or entities across the country to amplify its impact, be it other foundations, NCI, or the private sector. And this last slide just highlights, and these are just some pictures of people involved, but literally been so impressed by my colleagues around the country. We've had people put in hundreds and thousands of hours of personal work to make this happen. It's been a labor of love, and I just want to let you know that I'm very excited about where this is going to go. and I think it's going to really change the game for patients with pancreatic cancer. So I think I could answer a question or two. What we're going to do is we're going to take a question or two for each section, and then we're going to have a panel discussion at the end. So if anybody has a quick question, I'd be happy to address it. Yep. So the question is, there was a person who was young and actually had just delivered a child and was diagnosed, it sounds like at a very early age, with pancreatic cancer, and why couldn't they detect it? And we're going to talk, in the last session, we'll talk a little bit more about that, but that's one of the real problems of pancreatic cancer, is it does not present early. It's a very difficult diagnosis, and it often presents late. So while I have talked here very extensively about how we're going to change things in the clinical trial treatment space, I think equally, or perhaps even more important, is for us to develop more effective ways to detect the disease early. Yeah. So the question is, if your mother has pancreatic cancer, you'd increase risk for getting pancreatic cancer. And if it's okay, I'm going to defer that question to Jessica Everett, who's going to go over that in some detail in a little bit.